Good afternoon. My name is Zana Norton, and I am a biology and Spanish student working with Rema Sen under Dr. Lukasz Ciesla, and the title of my presentation is Diet-Induced Differences in EAGLEs Accumulation in Drosophila Melanogaster. In this talk, I will give some background on dietary implications, ages in EAGLEs, and Drosophila Melanogaster. I will then discuss my experiment, some preliminary results, and future directions. The focus of our research is on the dietary implications of the modern Western diet, in which highly processed foods are prioritized over nutrient-dense whole foods. Sugar is a large component of the modern Western diet, found in desserts, processed foods, and sugary beverages. The most common dietary sugars are glucose, fructose, and sucrose. Glucose and fructose are both monosaccharides, which means they consist of one unit of sugar. Sucrose is a disaccharide consisting of 1 unit of glucose covalently bonded to 1 unit of fructose. Liquid beverages have high sugar contents and are a staple in the modern Western diet. They are commonly sweetened with liquid sucrose and/or fructose, which dissolves more easily than crystalline sugar. This is the general pathway through which advanced glycation end products, or AGEs, form. The modern Western diet and its associated high sugar intake lead to hyperglycemia or high blood sugar. Chronic hyperglycemia leads to glucotoxicity, which is a broad term for various adverse effects. One specific mechanism of glucotoxicity is protein glycation, in which proteins in the body undergo a non-enzymatic, irreversible reaction with the sugar present in the blood. The products of protein glycation are called AGEs and inhibit various vital processes. Reactive AGEs may form via an oxidative pathway and tend to be more harmful. EAGLEs is an acronym for either advanced glycation or lipoxidation end products. These are a subcategory of reactive AGEs that form via oxidative glycation over a shorter period of time. This project focuses on exogenous EAGLEs, which form from external sources such as diet. Once EAGLEs form, they tend to accumulate in tissues. This causes oxidative stress, tissue damage, and inflammation. EAGLEs are implicated in neurodegenerative disorders such as Alzheimer's disease and Parkinson's disease. The accumulation of AGEs in pathological deposits associated with neurodegeneration worsens the course of these diseases. EAGLEs are also implicated in oxidative stress-induced chronic diseases. As I mentioned previously, hyperglycemia leads to oxidative stress and age formation. This can then trigger inflammation and cause atherosclerosis or diabetes. Drosophila Melanogaster is a species of fly commonly known as the fruit fly. It is considered a model organism for various reasons. For the purposes of our project, it is very helpful that Drosophila has previously been identified to form AGEs via the same mechanism as in humans. The figure on the right shows how the Drosophila life cycle occurs over a period of days as opposed to years, which allows lifespan and aging experiments to be conducted relatively quickly. They also produce many offspring, allowing for large sample sizes and the ability to repeat trials relatively quickly. The first aspect of this project is the survival assay, which measures the fly's mortality over time. We start by preparing a proprietary blend of fly food for the control group and adding sucrose or fructose as necessary to the food for the experimental groups. For a given trial, each experimental group consists of 3 vials with 10 flies each. We track the number of deaths per day and replenish the food every 4 days throughout the trial. These survival curves represent the probability of survival over a period of 20 days, with males on the left and females on the right. The different shapes and colors represent the various concentrations of sucrose or fructose. The key takeaway from the survival data is that there's quite a bit of variation from the experimental groups compared to the control groups. The second aspect of this project is the hemolymph extraction. Using a hollowed needle, a hole is punctured in the base of a small microfuge tube, which is placed inside a larger microfuge tube as shown in the figure to the right. The flies are anesthetized with carbon dioxide in the vial and then placed on a carbon dioxide pad. Using forceps, the head is separated from the main body segment and the contents are placed in the smaller of the microfuge tubes. The tubes are centrifuged and stored for future testing. So far, we have done a T-bars assay on the hemolymph, or a thiobarbituric acid reactive substances assay. This graph shows the highest sugar concentrations to the left, decreasing to the control groups shown on the right. This assay measures the concentration of MDA, or malondialdehyde, which is a marker often associated with the presence of AGEs. The highest presence of MDA was found in the control groups, which at first seemed counterintuitive. But my hypothesis is that the higher concentrations of MDA will be found in the tissues of the experimental groups at the sites of oxidative stress, as opposed to in the hemolymph. Now that we have established an assay to study the relationship between sugar intake and survival, we are working on biochemical assays of the collected hemolymph. This includes the aforementioned T-bars assay, mass spectrometry, and enzyme-linked immunosorbent assay, or ELISA. The goal is to identify a relationship between the lifespan data and protein glycation. We also plan to perform lifespan assays alongside pharmacological treatments. Thank you, and does anyone have any questions?
How would the pharmacological interventions or treatments treat the AGEs?
Yeah, so there's various mechanisms. The more common ones are going to prevent AGEs from forming in the first place, as opposed to getting rid of already formed AGEs in the body. So for example, we have drugs to treat hyperglycemia, which, as I mentioned before, is a precursor to age formation. So we can interrupt the AGEs before they form.
What made you pick this animal model as opposed to a different one?
Yeah, so for the other various reasons, it's very cost efficient, time efficient. And since there's the conserved processes of aging and the formation of AGEs, that's very helpful. If we chose a model such as the mouse or rat, one lifespan experiment would take over a year. So now we can have repeated trials and get lots of data to replicate over and over, where in other models we would take much longer to get the same results.
So are there any other factors besides diet that influence AGE formation?
Yes, there's many. So lifestyle is a huge component in the formation of AGEs. So things like smoking, alcohol consumption, and poor diet are going to promote the formation of AGEs. And then things like being active, having a good diet, and avoiding sugary and processed foods are going to prevent AGEs from forming.